So yesterday we were talking about evidence for evolution, and we left off with not yesterday, day before. We had left off on homologous structures and how very similar patterns of bones, for example, can do different functions. And then we also talked about vestigial structures and how vestigial structures don't serve any purpose anymore, but did at one point. Yeah, it is. And today I want to talk a little bit. We'll talk more about this in upcoming lectures, but there are these, we call these cladograms, and they basically are, well, I should go back to that previous slide there. They're calling them evolutionary trees here. They also call them cladograms. Don't worry about that right now because we'll talk about it later on. But these are their educated guesses, their hypotheses as to how related different groups of organisms are to each other. And so here's one where you can see that it's a whole bunch of different critters and how closely they are related to each other. Lungfish, amphibians, all the way through hawks and birds and other birds. And so we can use that. We can figure those out based on different things they have in common. For example, everything down here has an amniotic sac, doesn't have an amniotic sac, my bad. Whereas everything on the other side has it. That's just one example of that. Those diagrams are often based on things like fossil evidence. So looking at fossils and seeing, those species that we just looked at were living species currently, but sometimes they're based on trying to figure out how related a species is to one that is extinct, how closely related it is to something that's still living. So we've been talking about homologous structures, the idea that you can have a common ancestor based on having a similar bone structure, for example, one bone, two bone, many bone. But there's also a different reason that things can look alike. And that has to do with what we call convergent evolution. And convergent evolution doesn't necessarily mean, and usually doesn't mean, that there's a common ancestor. So it says down here at the bottom, it says convergent evolution doesn't provide information about ancestry. Because what can happen is you can have an environment, you can have two different groups of organisms very far apart from each other. But if they if they evolve in the same environment, they're going to have the same environmental pressures, and over time they will develop very similar features. And the example they show here on the next page is of, basically they're not really flying squirrels, but they are gliding squirrels. And we have a flying squirrel kind of in the eastern, southern United States. And it doesn't literally fly. It doesn't flap its wings and go for miles and miles, but it can glide from tree to tree. There's also in Australia a very similar organism called the sugar glider. The sugar glider, again, is basically a squirrel with little flaps of wings or little flaps of skin between its limbs that allows it to kind of glide between one branch and another. Okay. These are not, and they've done DNA testing on them and shown that they're not really very closely related. But if you just look at the surface, they look very much alike because of the fact that they both species grew up in heavily wooded areas where being able to jump from one tree to another was, beneficial. You splat on the ground, not so beneficial. So being able to get from one tree to the next was helpful. And so because the environments were the same, they look the same. That doesn't mean they are closely related.
Can I go take the test?
Yeah, you don't want to talk about sugar gliders?
I'm just worried about time.
Okay.
I do like sugar vodka. Do I need a pass or can I just get out?
Let me give you a pass because you're not signed up for today, and she may want you to sign up. And if I've got a pass, it's having you come from my class, then that will be the legitimate.
She's definitely gonna come back up.
But if I come back up, I don't have to take the test. Can all the juniors in here please take my Google form, to be honest? Because I really need the data for stats. Thank you. I'm actually begging.
Is it in the stream?
Yeah. Thank you.
I'm going to put on here, please register her. Thanks.
That's so kind of
So
you.
that should hopefully get you.
Great.
Right? And that
Yeah.
smile, that'll get you.
Thanks.
All right. All right. So, also, bats have, bats and birds, for example. Bats have wings. Birds have wings. They both fly. That's a very adaptive feature. But they are not very closely related to each other. That would be another example of convergent evolution, where you've got two groups don't look very much like each other, look a lot like each other, but when you actually dig deeper and you start to look at their DNA, they're not very closely related. So that's that convergent evolution as well. Again, with fossil record can show when a species went extinct, and then they can also show how a species changes over time. So here are some bones, ankle bones from different animals, dogs and pigs and deer, and then something called a pachycetus, or I don't know how to cetus, pachycetus. I think that's how you say it, which is a critter that we're going to see in a second that is extinct. You can see they're all pretty similar to each other. Fossils can document important transitions. We've seen cases where some kind of weird things. So, for example, I got some goo on my Chromebook. Sorry about that. Just trying to get it off. There we go. I want to go there though. I see cetaceans, things like whales. Do you know whales actually have hip bones? They have the remnants of hips. So the current thinking is that what actually happened, and you can see all of these things in yellow here that are highlighted in yellow. They are all related to each other. And the whales continued to live and continue to live today, whereas these guys didn't make it, but seem to have evolved into our current day hippos and deer. But they are all related to each other, according to looking at structural data, also looking at DNA. And you can see they do have the remnants of a pelvis, which suggests that they actually at some point were on land and the environment changed and they actually were better off in the ocean. So they kind of went back to the ocean. Fossils can show that the living cetaceans, which are just the whales, and their closest relatives, the even-toed ungulates. That just means that ungulates means they have hooves, and if they have 2 hooves versus 3, that kind of thing. They actually, let me just go back to the picture because I think it is easier to see it. So these whales were more closely related to these ungulates long ago than they are now to, say, the deer and the hippo. They're still related to the deer and the hippo, but they've gone farther away. They've evolved. Obviously, deer probably aren't going to do very well in deep water, right? And whales are. So over time, they've gotten farther and farther apart from each other. So they've diverged over time. This is the Is that the Pachycetus? Yeah. There's an even-toed ungulate there. You can see that does kind of look like it might be the predecessor to a deer. Yeah.
Wait, so that one, it like branched off in both directions to the hippo and to the whale?
No, I think, well, again, these are hypotheses, so we don't really know. So the idea is that the whales kind of stayed their course, and some of them branched off and became the ungulates. But it doesn't even really talk about why they had that pelvis. So there was something even before that we're not seeing. Anybody else smell like burnt toast?
Yeah.
All right. I wonder where that's coming from. I've been smelling various things in here that aren't from either that. Isn't it burnt toast you're supposed to smell right before you have like a stroke?
I thought it was burnt almonds.
Burnt almonds? Really? What does a burnt almond smell like? Like a burnt almond, yeah. Dummy. Of course, it smells like a burnt almond. Duh. All right. So next evidence is biogeography. And we talked a little bit about continental drift and how the Earth is made up of these little or these thin plates. And these thin plates move over time. And with them, they take all the soil and everything on top of it, including the continents. So at one point, we know that there was just one landmass, but it was humongous. It was called Pangaea. So Pangaea was this supercontinent that was made up of all the other continents that we have today. And over time, the movement of these tectonic plates have caused the continents to actually break off from each other and drift apart. And so now we have a lot of water in between. And so studying where, especially coastlines, because coastlines are representative of where continents used to be together. Like, for example, if you look at the coast of South America and the coast of South Africa, of Africa, you can see that they used to be butted up next to each other. And you can even see the outline of it. The shapes are very similar or complementary, like puzzle pieces. And you can also see that the species that are on those coasts are pretty similar. I mean, they're obviously very different than they used to be, but they look like they came from the same ancestor. An endemic species is a species that's not found anywhere else in the world, and you usually find those on islands because they're islands, they're isolated. It's not easy for something to move from one location to the next. Darwin suggested that a lot of the islands, the Galapagos Islands, for example, were once part of the mainland or very close to the mainland. And over time, they drifted farther and farther apart because of continental drift. And as they did that, they became isolated. Oh, really?
Yeah.
Did she say no?
She said no.
You gotta move.
You gotta move.
Okay, endemic species. So what he thought happened was over time, these islands got farther and farther apart from each other. And as they got farther and farther apart from each other, eventually they couldn't fly between islands. And one of the keys for making a new species is reproductive isolation. So if you're so far apart from each other that you can no longer interbreed, then that's going to create, over time, that will create populations that are going to become very different from each other. It's going to take a while for that to happen, but it will eventually happen. Okay, so Darwin's views of life are still being studied today, and we're trying to figure out exactly what's going on and why it is that some animal species are evolving the way they are and why they're the way some are staying kind of steadfast and not moving, et cetera, et cetera. These last few pages are some more direct observation or experimentations that, again, show support for the whole idea of natural selection and evolving over time. These are guppies. And guppies, if there's no predators, guppies will be very brightly colored. They'll have a lot of spots on them. and they'll be very colorful. But if they are in an area where there's a predator, they tend to have fewer spots, and they tend to be less colorful, because if you're colorful, you stick out. And if you stick out, you tend to get eaten. So there's a pool. There's an example here where they tested this in different pools. They had one pool that had this thing called a pike cichlid, and that eats the guppies. And in that case, you see that there's not many spots, because the guppies that have a lot of spots and are very colorful, they're the ones that get eaten. And then they transplanted them over into another pond that had these things called killifish, which were about the same size as the predator to the guppy. But the killifish doesn't eat guppies. It's just like, nah, you're cool. I'm not going to eat you. Okay, so it's the same size, so it controls for that size, but it's not going to actually eat it. And what they found was, over time, what happened was, I'm just making sure I don't get an email back that I'm a bad person. I'm sure I'm gonna catch it for what I just did.
Oh, yeah.
So, but I just don't care none. All right. So anyway, I just recorded that too. That's not good. Don't listen to what I just said. That was a substitute talking. Wait, everybody's in class. Oh, Evelyn's not. Evelyn. Anyway, so source population. Whatever the source population was, it had a muted, or it had less spots than when you transplanted it. When you transplanted it, you saw the number of spots here is on the side, area of colored spots. So they just became more colorful over time, because now they didn't have any predators. So the environment had changed, and as a result, the thing that's getting hunted, the guppies in this case, are also going to change. And that will then affect future generations of guppies. And so you get more colorful guppies over time. I think one of the last slides in here is not that one. Is on mosquitoes. And this is a big problem with pesticides. Mosquitoes reproduce pretty quickly. And we don't use DDT anymore to kill mosquitoes because it causes bad birth defects. But we still use it in labs to study things like evolution. And what they found was that if they expose mosquitoes to this pesticide called DDT, that the first exposure, only 4% of the population survived. But then if they let that 4% of the population continue to grow and reproduce and grow and reproduce, and then did it again after 8 months, 45% of the population lived. And then let those 45% reproduce, and then after 12 months hit it again with DDT. Now it was 77%. And this is just, this is very common. It's common for bugs, it's common for bacteria, it's common for viruses, that you get this buildup. If you don't kill off the entire population, it's going to come back, and now it's going to be resistant to whatever you used. And that's why it's important, again, that when you take those antibiotics, you take all of the antibiotic, because if you don't, you're going to get probably something that's resistant to that antibiotic, and it won't work, and you'll have to try something else. And usually that something else is more obnoxious and gives more side effects, and you just don't want to have to do that if you can avoid it. The other thing with bacteria and antibiotic resistance and stuff like that, I mean, we take for granted how important antibiotics are. I like to I like to listen to audiobooks that are apocalyptic in nature. Right, so the world kind of goes to heck, and then people have to live through it, right? Well, one of the things that goes in a post-apocalyptic world is antibiotics, and so they're talking about fighting between groups, and somebody gets cut, and that cut, it can get infected, and there's no antibiotics to deal with it, and there may not be even any antiseptic to treat it, like rubbing alcohol or anything like that. So then you end up with a situation where something that is just in our world would be so treatable that now without antibiotics, you're really in trouble. And we don't even have to think about post-apocalyptic futures. We just have to think about the fact that we, at this point, have so few antibiotics that work on some really nasty bugs, to see that we need to be careful about using our antibiotics. Okay, next chapter. This is on populations and how populations change over time in an environment. One of the very common things that, they're really subtle about testing you on this, but they like to test you on the idea that organisms don't evolve, populations do. Right? So you're not going to evolve over the course of your lifetime. You may pick up things that are environmental, but those things are not going to be passed on. So natural selection works on individuals, but it's only the populations that evolve. For example, after Darwin, many years after Darwin and his adventures on the islands, he had two scientists in particular, I can't remember their names now, who studied the Galapagos Islands, and they studied specifically ground finches on one of the islands called Daphne Major. You don't need to remember that. But basically it was the Grants, that was their names. It was a husband and wife team. And the Grants studied what happened to specifically just a bird's beak after years of drought. And they followed these cycles through, I don't know, like 10 or 15 seasons. And they found that after a drought, not during the year of the drought, but after the drought, so all of the new birds that would be produced the year after a drought, they tended to have beaks that on average they were much bigger. So here they're saying depth, but they're actually measuring like bird beak. That doesn't look like a beak at all. All right, let's try this again. That could be a beak, right? Oh, that's pretty good. Okay, I'm going to stop there because it's going to get so much worse. Okay, so that's my beak. So they're measuring from here to here. Okay, and so after a drought, the beaks would get substantially wider or deeper, as they called it, because the seeds were very dry, very tough, and very hard to crack. You couldn't get at the actual seed inside. Okay, and they also found that over time, if it was a wet year, the year after a wet year, the beaks got narrower because now you have to pick through the mud, right? You have to pick up little seeds through the mud. So it changed over time. Okay. So the finch, what evolved was not the finches themselves. Some of them died because they weren't able to crack those seeds, and some of them lived. But the next generation, then, the ones who tended to survive were the ones who had the deeper beak, and the population changed over the next few years. Microevolution is a term that we need to talk about. And this is, microevolution is when you have a change in the allele frequencies in a population over time. So what that means, allele frequencies. Now we're talking about what really changes over time is the genetic makeup of a population, right? Because our genes determine, genotype is going to determine phenotype, right? What you're going to look like is determined on what genes you inherited from your parents. Okay. So when we're talking about allele frequencies, we're talking about, do they have the dominant allele? Do they have the recessive allele? Do they have something in between? Sometimes it's not just dominant and recessive. Sometimes there's more than that. Changes in genotype or gene frequency or allele frequency are caused by three different things, and we're going to talk about each one of those in turn. Natural selection, genetic drift, and gene flow. The only one, however, that is really helpful for a population and is always going to cause the population to be better off is going to be natural selection. So that's the only one that's going to give adaptive evolution consistently. And we'll talk about genetic drift, and we'll talk about gene flow, and we'll talk about how that can actually harm a population, causing it to go backwards and maybe even go extinct. Again, kind of going back to why this can happen in the first place, there has to be variation. If a population doesn't have any kind of any variation, then that's just it's a non-starter. It's not going to happen. So that variation again is based on the DNA. So if the DNA is similar or if the DNA is different, are you producing the same phenotypes or are you producing different ones? I'm going through these kind of fast because it's a lot of common sense things that we've talked about before. Some phenotype differences are either you have it or you don't. So single genes, like you either have a widow's peak or you don't, which that I'm aware of doesn't cause any help to you in the environment. So there's no selective pressure to have a widow's peak or to not have a widow's peak. It's just either have it or you don't. Others, sometimes it takes 2 or more genes. Sometimes it takes many. Height would be an example of that. Skin color would be another one. There's a lot of genes that determine both of those traits. In horses, coat color. So this is a herd of horses that have very different coat colors because it's controlled by multiple genes. You can also measure something called heterozygosity. If you're heterozygous, what are your letters? Yeah, you'd have like a big A and a small a, right? You have different letters. So you could either give the dominant trait or you could give the recessive trait. So that's what heterozygosity means. It means how much of the population is heterozygous. That tends to give you more flexibility. If you get into a situation where you are just this, you can only give A. You can only give a dominant letter. If you are recessive, you can only give the recessive letter. That's it. That's all you're going to get. So not much heterozygosity. In other words. You can also look at the DNA sequence of individuals. A lot of times the DNA doesn't make a difference because the differences tend to be in non-coding regions. So those are in those intron areas that we talked about. And even when they are not in the intron areas, we talked about the idea that that third codon letter doesn't make a lot of difference. So, the amino acid CC, CCG, CCU, and CC, what am I missing? A. These all code for the same amino acid, which is proline. So it doesn't matter if that last one changes. And I kind of getting ahead of myself because mutations are coming up actually. So let's skip over that. So a source of genetic variation, some of it is sexual reproduction. So we talked about in meiosis, we talked about crossing over. We talked about independent assortment, about random fertilization. Those are all things that create variation. And just with crossing over alone, and it was in the billions, the different combinations that you can have when you look at crossing over and independent assortment, and you look at random fertilization. So lots and lots of variation from that. You can also get it from mutation and from gene duplication. So again, if you have a point mutation and I go from CCA or CCC to CCA, I'm still going to get the same amino acid. Okay, so a lot of point mutations don't do anything, or it's in an intron region that doesn't even get used. Effects of point mutations can vary. They can be harmful. Harmful mutations can often be hidden, though, because if it's a recessive allele and that mutation's in the recessive allele, you have to have 2 copies of it to get it. So if you only have one of it, it can be hidden. So somebody who's heterozygous may pass it on, but they're not going to show it. In general, though, mutations tend to be neither harmful or helpful. So they have neutral variation. So this is where they don't give you any advantage or disadvantage. They just are. Got it. It's a change in the letter, but other than, for purposes of saying it's different, in terms of advantages, there is no advantage, there's no disadvantage. Altering the chromosomal or altering the chromosomes tends to be more important, more detrimental. So if you delete a chromosomal region, that can be an issue. If you so if you lose a chunk of DNA, lose a chunk of a chromosome, that tends to be harmful. However, if you duplicate, sometimes that happens, where you duplicate a chunk of DNA. That tends to be less harmful. And a lot of our DNA, if we go in, we can actually look at our DNA and we can find that, it's not what I wanted. A lot of our DNA is duplicated. So we don't just have one gene for eye color or one gene for, a good example would be for, for some enzyme that's responsible for something in cell respiration. We might have 20 or 30 copies of it. The more copies of a gene that we have, the more cycles of meiosis it's gone through, the more time it's been passed down from generation to generation, and the older it is. So a cell respiration gene, which pretty much everything needs, would have a lot of copies. And something that it's like, oh, they discovered this gene and haven't seen it before, probably that one there isn't very many genes. There might only be 2 or 3 copies of that gene because it's a fairly new one. Another example of that is if you look at humans have, there's an odor detecting gene. I don't know what it's called. Humans have 300, don't have to remember the number. Humans have 380 and mice have 1,200. Okay. So they obviously went kind of in a different route, and smell became much more important to mice than it does to human beings. I mean, we smell pretty good, but not as well as rodents do in general. Even though we get mutations, we don't get a lot of them. They're pretty rare because of the fact that there are DNA polymerases that will scan the DNA and check to make sure it's accurate. And then there's like P53 that halts the cycle. So if something's not working right, it'll halt it, make it fix itself before that cell's allowed to go on. In average, there's one mutation in every 100,000 genes per generation. So generation means every time a cell divides. It's pretty good. Mutation rates are lower in prokaryotes. However, even though they're lower, they have shorter generation times. So, you can have, I don't even know the math, but you can have lots and lots of bacteria in a 24-hour period of time. So even though they have fewer mutations over a 24-hour period, you could have so many generations that the mutations would start to add up after a while. Even higher than that is viruses. Viruses can copy themselves very quickly if they've got a host that they can do it in. And they don't have any way to check that their DNA is correct or their RNA. And so as a result, you get viruses mutate the fastest and can be the most deadly because they're going to change over time so quickly. And we saw that with COVID. All right. I'm going to stop there because I know y'all are